In this video, I will go over the function of different cellular structures. So the first structure is the smooth endoplasmic reticulum, which is required for detoxification. So we have the smooth endoplasmic reticulum, which detoxifies metabolites through two different phases. Phase 1, which involves oxidation, reduction, as well as hydrolysis, all of which are done by the P450 enzyme. And so it will yield metabolites that are polar and water soluble. And then through phase 2, these metabolites will undergo conjugation. And examples of conjugation include glucuronidation, sulfation, or acetylation. And by doing that, it will yield highly polar inactive metabolites. Now, with aging, cells become less efficient in detoxification, and it's due to the dysfunction of the P450 enzyme. So just know that with aging, phase 1 metabolism is the first to be affected. The next structure is the nucleus. So here we have the nucleus, and then it has two forms of chromatin, the heterochromatin, which is dense and is expressionless, and then we have the euchromatin, which can express different genes. So euchromatin is required for DNA replication and the production of mRNA through transcription, and then mRNA will exit the cells, and through ribosomes it will form proteins. And then the next structure that we have here is the nucleolus. And the nucleolus will produce the ribosomal RNA, and then the ribosomal proteins that were made in the cytoplasm will be transported back into the nucleolus. So ribosome proteins will also be transported back into the nucleolus, and then all these structures will be assembled together. So 40S and the 60S subunits will be assembled with the rRNA all together. And then all these structures will be then exported through nuclear pores back into the cytoplasm. Now, once in the cytoplasm, mRNA will come right in between and will be producing proteins through the process of translation. The next structure is the rough endoplasmic reticulum. So here we have the rough endoplasmic reticulum, and you can see that the ribosomes are attached here to the membrane here. So I would like you to know that we have two forms of ribosomes. One is the free ribosomes, and the proteins that are produced by free ribosomes will remain in the cytoplasm. And then two, we have ribosomes that are attached to rough endoplasmic reticulum, and proteins that are made by rough endoplasmic reticulum are destined for lysosome, Golgi, smooth endoplasmic reticulum, cell membrane, as well as secretion. Now, in addition, rough endoplasmic reticulum has one other function, and it's N-linked glycosylation of proteins. So a glycan, also known as a sugar molecule, will be added to the nitrogen of the asparagine, and through that, glycoproteins will be made. And there are two main purposes to the formation of glycoproteins through N-linked glycosylation, and the first one is to stabilize proteins. So if you somehow interfere with N-linked glycosylation, most of these proteins will undergo degradation. And then the next function that may be present is for protein folding. So sometimes N-linked glycosylation helps the proteins to fold into the correct structures. The next structure is the Golgi apparatus. So here we have the Golgi apparatus, and it has two sides, the cis side and the trans side. So the way I memorize it is that cis is the receiving part, while trans is the shipping part. So organelles will be shipped out of the trans section to different parts of the cells. So there are two main functions that are associated with the Golgi. The first one is O-linked glycosylation, And then the second one is protein sorting. So Golgi apparatus determines where proteins should go to. And then I would like you to know that proteins to which mannose 6-phosphate is added are all destined for the lysosome. So just keep that in mind. Any protein in the Golgi apparatus to which mannose 6-phosphate will be added will go to the lysosomes. The next structure that I'm going to cover is the lysosome. So here you can see that we have a lysosome right here, and then we have different organs, like here we have mitochondria inside that are being digested. Then the next structure is the peroxisome. So here we have the peroxisome, which has two main functions. So the first function is detoxification of hydrogen peroxide. So detoxification of hydrogen peroxide. And then the second function is for the metabolism of long fatty acids and amino acids. And then the final structure that we have here is the mitochondria. So here we have the mitochondria, and the way you can recognize these on these images is all these cristae that is inside the mitochondria. So here we have the cristae, and then cristae right here. And then the function of mitochondria is ATP/energy production in cells. And that concludes our discussion.